Very good evening, my dear friends. So this is the second session of our PG competitive exams botany online coaching. Welcome to today's session. So I would like to introduce one of the eminent resource person, Dr. A. Madhuri Madam Garu, Assistant Professor, Department of Microbiology, Government Degree College for Women, Autonomous, Begumpet, Hyderabad. Madam has huge experience in the field of microbiology, at the same time the plant sciences. So we will going to invite our eminent research person, Dr. A. Madhuri Madam Garu. Please share your screen. I have given to you, Madam. Please share your screen and start the session. Madam, click on slideshow. Madam, click on slideshow.
Yes, sir. Some technical problem.
Plant science lo bacteria
Is this clear, sir?
bacteria గురించి అయితే ఉంది bacteria
మేడం మీ దాంట్లో మీరు రెండు ద్వారా ఉన్నట్టున్నారు, ఒకటి మ్యూట్ చేస్తే మీరు క్లియర్ వస్తుంది. My dear friends, Madam is going to start the session.
One second, sir. I am trying.
మేడం మీరు ఒకదాన్ని మ్యూట్ చేయండి, మీరు ఒకటి మ్యూట్ చేసి ఒకదాని ద్వారా మాత్రమే మాట్లాడండి. Is very expert in microbiology, so Madam related field and the Department of Microbiology, so that's why Madam has given a wonderful session. Will give a wonderful session. My dear friends, take notes during the class time, and if you have any doubt, please post the questions in the chat box, or otherwise you can direct interact with our today's resource person. Now, not only today, each and any class, whatever you have any doubt, which type of you doubt, please you clarify during the class hours, no problem. Otherwise, please mute yourself and listen carefully. Okay, Madam is going to share the screen. Please wait for 2 minutes.
Myself, Dr. A. Madhuri, Assistant Professor of Microbiology. Today, I'm here to share information about ultrastructure of bacteria. Am I audible to all of you?
Yes, madam. Yes, please you proceed.
Yeah. Like, can the students talk to me, sir, during the lecture?
Madam, 18 students join.
Okay.
మీ వాయిస్ కొంచెం తక్కువ వస్తుంది అంటున్నారు, కొద్దిగా బిగ్గర్ గా
వాయిస్
enhance your voice louder.
Okay.
Thank you.
Okay, sir. So as we all know, bacteria are microorganisms. Why we call them as microorganisms, ma? Because they are very smaller in size. That is why we are calling them as microorganisms. And there are different types of microorganisms, but today we are focusing on bacteria, right? Now, bacteria, we call them as prokaryotic microorganisms. What do you mean by prokaryotic? Pro means primitive, karyon means nucleus. If the nucleus is primitive, means if the nucleus is not well developed or if the nuclear membrane is absent, such microorganisms can be called as prokaryotic microorganisms. Then, bacteria are usually single cell and has a DNA in the center of the cell, which is not bound by nuclear membrane. The major difference between a prokaryotic cell and an eukaryotic cell is the difference or absence or presence of nuclear membrane. If the nuclear membrane is absent, you have to call them as prokaryote. If the nuclear membrane is present, you have to call them as eukaryote, right? And other characters of prokaryotic cells are they are lacking membrane bound organelles, means the organelles like endoplasmic reticulum, mitochondria, chloroplast, Golgi complex, all these organelles are absent in prokaryotes. And bacteria also have plasmids. What are plasmids? These are extra chromosomal genetic elements. And so, where these bacteria are found, they are omnipresent, means they can be present everywhere. They are the earliest inhabitants of the earth, means from many years they are present on the earth, approximately 3 into 10 to the power of 9 years old organisms. And they can be seen in soil, water, air, hot springs, earth's crust, anywhere you can see these bacteria. And they can show symbiotic and parasitic relationship. What is symbiotic? Sym means together. SYM means together. Bio means life. The organisms which are living or leading the life together are called as symbiotic. And parasitic means you all know they are dangerous or they can cause diseases. And you can be surprised if you take a gram of soil, you can find 40 million bacteria in that 1 gram of soil. Then, as we humans, we are more like concerned about our hygiene, cleanliness, but on our body, it will be 10 times more bacteria than the cells. Okay, so these are present everywhere, and we are not free of microorganisms. They are present on our body and in our body also. Next. So why we have to study about bacteria? As they are very small organisms, they can be used in research fields, and they are helpful in fermentation processes. They are helping in carbon cycle, nitrogen cycle, and they can be given as food for animals or humans in the form of single cell proteins. And they are also helping us in cleaning the environment. Okay, so bacteria are very important and very useful also. So see the scale here. You can see this is the range of light microscope. This is the range of eye. Means to this level we can see with the eye, means just below 1 mm, little less than 1 mm. Okay, and beyond this you can see through light microscope, and beyond this you can see through electron microscope. Mostly the bacteria will be in the range of micrometers size. Next, coming to the size of bacteria, spherical bacteria can be 0.5 to 2 microns. Rod shaped bacteria can be 1 to 10 micrometers. Means here you can understand microorganisms or bacteria are having different shapes. They can be round shape or rod shape, and they are also having different sizes. Okay, this Epilopicium fissuloni, this is the size of this is around 600 micrometers into 80 micrometers. This is the biggest cell. Likewise, mycoplasma is the smallest cell. And coming to the shapes, as we have discussed, there are different shapes. Cocci. Cocci means round shaped cells. See this figure? This is monococci, diplococci, two cells together, staphylococci, grape bunch. Your examination. In your examination, you can be asked about the shapes of bacteria. Okay, so this slide is more important. Staphylococci is grape bunch arrangement, and streptococci is a linear arrangement of cells, and sarcina is a cuboidal arrangement, and tetrad is a 4 cells together. And these are the examples. You can be asked which bacteria is showing which kind of arrangement. So you have to remember these examples. Okay, Diplococcus means 2 cells together. Example is Diplococcus pneumoniae. Similarly, micrococci, single cocci, Micrococcus roseus, Streptococcus, Streptococcus lactis, Staphylococcus aureus. Tetrads can be seen in Aerococcus and Pediococcus. And sarcinal arrangement, cuboid arrangement can be seen in Sarcina lutea. So these are general examples. Apart from this, many more cells can show this kind of arrangement. Next is Bacillus. Yeah, some lines are coming, ma. I am not able to avoid this, but my monitor is very clear. I don't know why it is showing like this.
Please, madam, proceed. No problem.
Okay, so coming to the Bacillus, Bacillus means rod-shaped bacteria, and this shape, this is the intermediate between cocci and rod-shaped bacteria, first cell. Okay, this is Coccobacillus. Name is Coccobacillus, and second is single cell, which is of rod-shaped, and then Diplobacilli, and this is palisade arrangement, like matchsticks will be there now in the matchbox. Similarly, these cells are arranged in linear arrangement. This is called palisade arrangement, and the next one is strepto arrangement or streptobacilli, and these are the examples. Okay, you have to remember each one example. Next, Vibrios is comma shaped. Comma shaped cells. See the shape. This is something like of comma shape. Example, best example is Vibrio colorae. And next one is Spirillum. It is a helical cell. Example says Treponema pallidum. They appear like corkscrew, some kind of screw shape it will show, and they are called as spirilla. Not only these, say now you can like understand what kind of shapes we have studied till now. We have studied about round shape, that is cocci, rod shape, bacillus, and comma shape, that is vibrio. Okay, apart from that, there are other shapes. What are the other shapes? This is filamentous, candidatus savagilla. This is sorry, irregular shape or pleomorphic. Many shapes can be seen. This is mycoplasma, and this is, you can see, square shaped cells or rectangular cells. Here this can be seen in Halarcula, Valismotis, and star shaped cells can also be seen. Example is Stella. Coming to the ultrastructure, as we have less time, I'll be moving a little fast, but you have to remember only basic things will be asked for your subject from microbiology. Okay, so this is the important diagram. You have to understand the construction of the bacteria, how it looks. Starting from the outer side. Okay, these are flagella. These are the flagella, and these are pili. Okay, and below that you have capsule. Below the capsule, cell wall. Next, cell membrane, and then you have inclusion bodies. Then this is DNA, and these are plasmid. You can see some round structures are there. These are plasmids, extra chromosomal elements. Okay, and the small purple dots are ribosomes, which help in protein synthesis. Okay, so this is the construction of the bacterial cell. Pili, capsule, cell wall, cell membrane, DNA, inclusion bodies, ribosomes, and plasmid. These are some of the important parts of the bacteria which you have to remember. Apart from this, you can also see mesosomes. So first one is flagella. Flagella, long thread-like structures. You have seen now in the previous slide. These are flagella. Okay. So they are helpful in motility. They are made of proteins called flagellin. They are useful as taxonomic tools. Means you can classify the bacteria based on flagella, and it consists of three parts: basal body, hook, and filament. The main function of flagella is motility. This is the structure of the flagella. Uh, this much depth is not required for you, but there will be rings. This is the gram negative cell wall, and this is the gram positive cell wall. Where peptidoglycan is more, this will be gram positive cell wall, and this will be gram negative cell wall. In gram negative cell wall, the arrangement of flagella is somewhat different, where you can see four rings: L ring, P ring, S ring, and M ring. And this is the plasma membrane. This is the cell wall. Okay, right? So this is the basal body. Here this is the hook, and after hook there will be a filament. This is the ultrastructure of flagella. Next, different types of arrangement. If you have single flagella, it is monotrichous. If you have a tuft of flagella at single end, it is called as lophotrichous. Here you can get bits, questions. Tuft on both sides, amphitrichous, and on all the surface of bacteria, it is peritrichous. And if you don't have flagella, it is called as atrichous. Okay. Next, monotrichous example: Pseudomonas aeruginosa, lophotrichous Pseudomonas fluorescens, amphitrichous alcaligens, and peritrichous is Salmonella typhi. Coming to the pili. Mostly they are seen in gram-negative bacteria. They are made of protein called as piline, and they are smaller than flagella, and they are more in number, and they are controlled by genes on plasmids. Like we have seen, plasmids are extra chromosomal genetic elements. So the genes responsible for pili formation are present on the plasmids, and do not help in motility. They can be removed mechanically, and there are different types of pili. What are the functions? They have antigenic property. Means when a bacteria enter into our body will respond against that bacteria. Okay, means our body will produce antibodies towards that bacteria. So the pili is acting as an antigen to which our body will produce antibody, and it will try to kill the bacteria. And fimbriae agglutinates with blood cells, blood grouping, if you see. Okay, their clotting can be seen. So that agglutination reaction can also be seen by pili, and it affects metabolic activity. Sex pili transfer genome by the help of conjugation. You might have studied about conjugation in your lower classes, where genetic material or plasmids can be transferred from one cell to another through conjugation tube formed by pili. Some pili are used for attachment for pathogenic bacteria. When they enter into our body with the help of pili, they will attach to our internal layers, mucous membranes. Next is capsule. Capsule is the outermost layer of the bacteria. This is the slimy or gummy material. It is also called as glycocalyx. If the material is bound tightly, it is called as capsule. And if it is bound loosely, it is called as slime layer. This you can remember. What is the difference between capsule and slime layer? Capsule is something which is tightly bound, and slime layer is something loosely bound. They are helping in pathogenicity. Means when the bacteria enters into our body, it will escape from our immune response by having capsule. And polysaccharides, it can be made of polysaccharides or polypeptides. Polysaccharide capsule is present in Streptococcus and Xanthomonas, and polypeptide capsule is present in Bacillus species. So this is the capsule. When you stain, this halo or clear space around the cell can be seen. Okay, so this is capsule or slimy material. Just now we have discussed it helps in pathogenicity to cause the disease, and it helps from drying. It helps the cell from desiccation, means it will not allow the cell to dry, and exclude bacterial viruses and toxins, means it will not allow virus to attack the bacterial cell. It will not allow virus to penetrate the bacterial cell. And for attaching to streams, plant roots, so for attaching to substrates, any substrate, and it is species specific. It is acting as buffer between cell and external environment, means it is protecting the cell. Next, below the capsule we have seen, next is cell wall. It is protecting the cell. It is giving the shape to the cell, and it is also protecting the cell from osmotic pressure. Actually, it is the strong layer which is present in the bacteria, and thickness is around 100 to 250 angstroms. There are two types of bacteria based on cell wall. They are gram positive and gram negative. Most important component of cell wall is peptidoglycan. Okay, and in peptidoglycan there will be backbone is there, tetrapeptide side chain is there, peptide inter bridge or cross link will be there. Okay, so here just I'm skipping this because this much depth is not required for you. Even then I'm just showing the structure. This is peptidoglycan. In peptidoglycan there will be two units, NAG unit and NAM unit. Okay, NAG is N-acetyl glucosamine, NAM is N-acetyl muramic acid. These are linked together like bricks in the wall. Okay, they are linked together. They are present in layers, and the opposite, what do you say, units of adjacent membranes are linked by peptide bridge. Okay, so this is the structure of peptidoglycan. And this can be important for you. This is gram positive cell and this is gram negative cell. This difference you have to understand. In gram positive cell, there are many layers of peptidoglycan are there. In gram negative cell, only one or two layers are there. This is the major difference. Okay. What is the difference between gram positive and gram negative cell wall? Peptidoglycan content is the difference. Okay. It is more in gram positive, less in gram negative. Right. Then, what are the other differences? There are teichoic acids in the gram positive cell wall. Here there are no teichoic acids in the gram negative cell wall. Apart from that, on outer side of peptidoglycan there is a lipopolysaccharide or outer membrane is present in the gram negative cell wall. Okay, and this O antigen, all these are acting as a antigens. Next. Wall. It is cell wall has relatively low amino acids and all the amino acids are present, means amino acid content is high in gram negative cell wall, lipid content is high in gram negative cell wall. Muramic acid means peptidoglycan, other name for peptidoglycan is muramic acid. It is more in gram positive cell wall, and teichoic acid is present in gram positive cell wall, absent in gram negative. Lipopolysaccharides are absent in gram positive and they are present in gram negative cells. Polar flagella, here they are absent and here in negative they are present. Sensitive to penicillin and sensitive to streptomycin. Based on this cell wall structure, they are showing sensitivity towards different antibiotics. So what is the purpose of cell wall? Main purpose is to give the shape. When you are saying a particular bacteria is round, a particular bacteria is rod shaped, the main reason for that shape is the presence of cell wall. Okay. So it is also a protective barrier which is helping the cell to survive, and it is having LPS, lipopolysaccharide in gram negative cell walls we have seen now. So there lipopolysaccharide is there, and matrix proteins, they are acting as receptors for nutrients, polysins, bacteriophages. Means there is a lipoproteinaceous membrane and protects the cell protoplast from mechanical damage. If you remove the cell wall, the cell cannot be protected against osmotic pressure. There can be enzymes responsible for respiration, photosynthesis, and lipid synthesis on membranes. Next, plasma membrane, it is present below the cell wall and it is having 5 to 10 nanometers thickness. It is a lipoproteinaceous membrane. It is having lipids and proteins. 70% proteins and 30% lipids are there. They can be of two types. Proteins are of two types, peripheral proteins and integral proteins. And coming to the it is a continuation. So thin lipoprotein below the cell wall. That's all. Here you can see this is integral protein. It is present throughout the membrane, and these are this is peripheral protein. Okay, it is present on one side of the membrane. Hopenoids in bacteria are sterol-like molecules present, and the functions of plasma membrane is they help in transport of nutrients. They contain enzymes for different pathways and electron transport, oxidative phosphorylation, everything will happen on the plasma membrane. And it is also gives attachment sites for chromosome and plasmid replication. Next, mesosomes. This can be of important point for you. Mesosomes are invaginations of plasma membrane. You have studied about plasma membrane just now. Okay, so if that membrane forms some invaginations, some foldings, that is mesosome. Okay, and mostly it is seen in gram-positive bacteria. Scientists think that it is helping in cell wall formation during cell division or chromosome replication, or it can be a site of oxidative phosphorylation. And similar folds in bacteria can be seen when antibiotics or antibacterial peptides are acted upon bacteria. Means you can understand previously scientists used to think that mesosomes are part of bacteria, but now they have some other conclusion like mesosomes are not part of bacteria. They are formed under special conditions. If you are putting some pressure on the bacterial cell, some kind of pressure, it can be of antibiotic or some chemicals, whatever, then these mesosomes are formed. It is a like recent findings by the scientists. Next is ribosome. Ribosome, you know very well. These are protein synthesis. They help in protein synthesis. And these ribosomes, they have 60% proteins. RNA is 60% and protein is 40%. What kind of RNA is present here? It is ribosomal RNA, that is rRNA. It is 60% and protein is 40%. Prokaryotes, what type of ribosomes are present? It is 70S type and eukaryotes 80S type. If you have 70S type, it can be having two um, subunits. Okay, one is 50S and the other one is 30S. What does S stands for? It would be better if I can interact with the students. S stands for Swedberg units. Okay, 70S breaks into two units, that is 50S and 30S. If you add these two, it won't be equal to 70S, it will become 80S. Okay, so whatever, when you divide, when you break the components, this is the Swedberg units given by these subunits. Okay, so 30S will have 16S rRNA and 21 proteins will be there, and 50S will have 23S rRNA, 5S rRNA, and 31 proteins. Okay, so now we are, as we are talking about bacteria, we are, let us ourselves constrained to this prokaryotic ribosome, right? And this 16S rRNA, which is present in 30S subunit, is important for tracing the evolutionary relationship. Okay, this can be asked. So just remember 16S rRNA. Diameter is of ribosome is 200 angstroms, and polyribosomes means if the ribosomes are attached together, then it is called as polyribosome. So this is the subunit, 30S subunit, 50S subunit, and this is 70S ribosome where two subunits are together. Coming to the nucleoid, it is the central part or DNA, which is most important part of the cell. It is having single DNA molecule, which is double stranded and circular in nature. You can stain the nucleoid or DNA by using Fuelgen stain. DNA is attached to the cell membrane, and nucleoid it will have 60% DNA, 30% RNA, and 10% protein. Proteins, they resemble histones. They are called as HUNS and DNA binding protein too. And they are lacking introns and histones. They are lacking introns and histones. What do you mean by introns, ma? Exons are coding sequences. What are introns? Introns are non-coding sequences, right, which can be seen in eukaryotic genome. But these introns are absent in prokaryotic genome. So if you see the bacteria size is very small, but the DNA, it is having lengthy DNA. So it will get coiled and then it will get integrated or present in the small place of the bacterial cell. Okay, so this coiling is called as when the coiling undergoes again coiling, it is called as supercoiling. So this is the method how the bacterial DNA will coil itself. And E. coli DNA, E. coli is 2 to 6 micrometers in size, whereas the DNA present in E. coli is around 1400 micrometers. Okay, so see the length, how much DNA, how much lengthy DNA it is able to accommodate. That's such a small cell, it is accommodating a big DNA molecule. Next, plasmids. These are extra chromosomal DNA elements we have seen in the ultrastructure. Their size can vary, their number can vary, and they help in presence of antibiotics or stress. Means they will have some special genes, and this can be transferred from one bacteria to another by different methods. And 1 to 100 copy number means the number of plasmids present in a cell is called as copy number. Okay, and episomes of plasmids, these are all different types of plasmids. Are plasmids called plasmids based on the function? They can be of different types of plasmids. Coming to the bacterium, here, if this is the main genome or main DNA, this is called as plasmid. Okay, here in electron microscope picture, you can see the small structures, these round structures are called as plasmids. Next, functions of plasmids, as I told you, they will have separate genes or special genes. They can make toxins with their genes. Or they can help in resisting environmental factors, or they can resist various antibiotics, and they can use unusual chemical compounds. Means in the starting slide, we have talked about using bacteria for environment cleaning. Okay, so how they are cleaning? They are having some separate genes, special genes, which can help in breaking the toxins or breaking the pollutants. They are also having storage granules, polyphosphate, sulfur, glycogen, all these can be stored. Sir, how much time is left? So gas vacuoles can
8
also be
minutes.
there. Okay.
8
So this gas vacuoles means um, they are helpful in buoyancy of the cell, floating of the cell for photosynthetic bacteria and cyanobacteria. And carboxysomes are they help in storage of this enzyme. RUBP carboxylase enzyme is stored, which helps in carbon dioxide fixation. It can be of alpha types or beta types. And magnetosomes, these these are like magnets present in the bacteria. If you can see here, they have this iron particles, Fe3O4 magnetite material is there. They are such, that is why they are called as magnetosomes. And example is Aquaspirillum magnetotacticum. They help in magnetotaxis, means according to the magnetic field they can move. And this magnetosomes can also be seen in heads of birds and dolphins, which they help in guiding the movement of these organisms. Tuna, green turtles, all these they are having this magnetosomes. Okay, so this electron microscopic picture, magnetosome chain, small magnets. Okay, they can be present in bacteria also. Endospores. What do you mean by spore? Basically, it is a structure which is helpful in resisting unfavorable conditions. When the bacteria spore is formed, it is helpful in resisting unfavorable conditions. Mostly, it is formed by gram-positive cells, and examples are Bacillus, Clostridium, etc. They are helpful in resisting heat, radiation, chemical disinfectants, and drying or desiccation. And they survive in boiling water for several hours. So if you boil the water and if you think we have killed the bacteria, sometimes it may not happen due to presence of spores. If spores are present, then these spores will not get killed in boiling conditions. After the water gets cooled, they can germinate and they can again cause the damage. So spores, their shape can be oval, ellipsoidal, or spherical, and the shape position of the spore can be terminal, subterminal, or central in position. Terminal means towards the end of the cell. Subterminal means towards one end, and central means in the center. And DNA, RNA, enzymes, calcium dipicolinate. So what this spore will have? They'll have DNA, they will have RNA, they'll have enzymes, as well as some calcium dipicolinate will be there, which is the main component which makes the spore harder to cope up with the unfavorable conditions. And 10 to 30% water means water content comes down in the spore. And proteins, so small soluble proteins are there. SASPs, they are called as SASPs, and these SASPs will bind to DNA to protect the DNA from UV light, drying or desiccation or heat. And SASP serves as carbon and energy source also. If the cell is in danger, they can also serve as carbon and energy source for the spore. So ultimately, spore has to survive the unfavorable conditions. So this is the endospore. And the name itself, endo means within the cell, exo means outside the cell. So within the cell, spore is formed. That is why we are calling it as endospore. And this is the parent cell. Okay, this is the central spore. This is the terminal spore. This is the subterminal spore. And these are the walls, exosporium, then spore